We're so happy that you all could make it here to learn about some of the exciting work in Parkinson's disease we have going on at the University of Buffalo. I'm Jean Witowski-Wendy, and I'm Vice Provost for Strategic Initiatives at the university. The strategic plan that our current president and former president put in place invested in areas of strength of research at our university. And some of those strength areas are what we're going to present today. The strategic strengths included investment in the areas of health and wellness, and also in areas of molecular recognition, in biologic systems, and bioinformatics. And today on our speakers list, we have both a basic scientist, Dr. Feng, who's going to talk about his work in molecular recognition area, and Dr. Tom Gattuso, who's going to present a clinical perspective on Parkinson's disease. I just am here to welcome you. I also want to introduce a few important people that are here with us today. First, Dr. Michael Kane, who's up front, who is our Vice President for Health Sciences. He's also the Dean of the School of Medicine and Biomedical Sciences, and he coordinates two of our strategic strengths as a dean in charge of them. So I welcome Michael and also welcome him because he's a great sponsor of this program as well as our research in these important areas. I also want to introduce Dr. Ken Blumenthal, who is our Senior Associate Dean for Research and Graduate Education in the medical school. He's also Professor and Chair of the Department of Biochemistry, and he leads the Faculty Advisory Committee in Molecular Recognition. Who is not with us today is Bill Reagan, who is the Director of the Office of Special Events, and I have to have a special thanks to Bill, who really worked to make this happen. This is a first event where we try to bring together both the research at our university and our distinguished speakers, and obviously it's been a success so far based on the attendance. So I want to thank Bill Reagan, his office, and also the staff from the Center of the Arts. They've done a wonderful job trying to accommodate this unexpectedly large crowd. I also want to recognize Michael Torcell from my office, who's helped to coordinate this. The event today will be moderated by Dr. Blumenthal. He will introduce the speakers, and we're hoping that between the two speakers, we'll still have 15 or 20 minutes for question and answer, which Dr. Blumenthal will moderate. We're hoping to get out of here promptly at 7:30 for those of you that are walking over to Alumni Arena to see the event with Michael J. Fox. It should be more than sufficient time, so we'll try to keep on time. Again, I want to thank you all for coming. I hope you enjoy this wonderful program, and Dr. Blumenthal.
Good evening, everyone. I'd also like to thank you all for coming. So it's my pleasure tonight to be able to introduce our two speakers, and I'll just give you a little bit of a biography of both of them. Jen Feng joined the faculty here in the Department of Physiology and Biophysics about 11 years ago, and came as an assistant professor. Based on his achievements, he has risen fairly rapidly through the ranks to the point where he's now a full professor. Jen received his bachelor's degree in biochemistry in China, and then came to the US and received a PhD also in biochemistry. And parenthetically, I should say that as the chair of biochemistry, I have to compliment him on his choice of discipline. Received his PhD in biochemistry at the University of Tennessee College of Medicine. He then moved to Rockefeller University, where he worked in the lab of a Nobel laureate, Dr. Paul Greengard. And while at Rockefeller, he developed an interest in a newly discovered protein, which was called Parkin. Now, Parkin is a protein whose precise cellular function is not really known. But it is known that mutations in the gene that code for Parkin lead to the death of certain neurons and an inherited form of Parkinson's disease. So there's a clear relationship between this gene and gene product and the disease. Jen and his students have discovered several critical functions of Parkin in human midbrain neurons and are working on identifying novel drugs that are capable of mimicking the beneficial effects that Parkin has in promoting neuronal survival. Dr. Feng's research team is supported by multiple grants, including grants from the Michael J. Fox Foundation, from the National Institutes of Health, and from the Empire State Stem Cell Board. And you're going to hear some information about the work that's supported by the stem cell grant. After his talk is over, Dr. Tom Gattuso will provide a clinical commentary, clinical perspective on the implications of the work that's being done in the Feng lab. So Dr. Gattuso got his undergraduate training at Cornell and then did his MD at UB and got the degree in 1996. He then went off to Rochester for a residency and a fellowship in movement disorders, came back to Buffalo as an assistant professor of neurology in 2003. He's been the director of the Movement Disorders Fellowship Program here since 2007. Won the Neurology Residence Outstanding Teacher Award in 2009 and is the author of several original research papers and clinical studies on analysis of Parkinson's-directed drugs and on genetic markers for the familial form of the disease. His research has been funded by the NIH and by the National Parkinson's Foundation, as well as by industry-supported clinical studies. So with that, by way of introduction, I want to bring Dr. Feng up on the stage, and he will tell us about his recent work on new directions in Parkinson's research. Thank you all for coming.
Well, thank you very much, Ken, for the kind introduction. It's a great pleasure for me to be here to tell you our work, and hopefully we can inspire more work, and I think we need to work together to find a cure for Parkinson's. Can I have the slide? Okay, good. So we're trying to find a cure because right now the treatment for Parkinson's disease is not a cure. So I think the beauty of science is allow us to see the external world, including ourselves, with logic. And so when we analyze anything that moves, obviously, for example, if you look at this very beautiful formation done by the Navy Blue Angel team, you can, if you think about it, you can realize that these precision movement must be under some kind of control. A human obviously can do the same kind of movement, very beautifully manifested by Michael Jordan here. So the reason for this precise movement is actually the same principle. So we are controlled by a computer, by a very, very good computer, which is our brain. And this is actually the axons, so the major axons. These are the nerves inside our brain, show on a specialized form of magnetic resonance imaging. So our brain is actually a remarkable computer. It's actually now the best computer can perform computation up to 1,000 trillion times a second. And it's not nearly as it's not nearly as good as a human brain. So our brain has about 100 billion neurons. These are the nerve cells. And each of these neurons make, on average, about 1,000 connections with each other. These are called synapses. So it is the synapse that helps the nerve cells to make these neurocomputation. And everything we do, including the fact that I can stand here and speak to you, is because we have these neurocomputation taking place inside the brain. And one of the subsystems is actually this picture showing here. This subsystem controls voluntary locomotor activity. So for example, our locomotor movement, how we move, how we talk, how we control our posture. This is the function of this subsystem. So this system has two major parts, if you look at this picture. These are the neurons, nerve cells that produce dopamine. These cells actually send a very long projection into another part of the brain called the basal ganglia. So in here, there's a lot of there are a lot of neurocomputation going place. There are a lot of neurocomputation taking place every second to make sure that we have a well choreographed sequence in our brain that controls our locomotor activity. So if you look at Parkinson's disease, what happened is that this whole system is not functioning properly. So the system is, so the centerpiece is actually the neurocomputational network in the striatum. Exactly how these neurocomputations take place, we're not exactly sure, but we're absolutely sure is that these nerve cells need to have dopamine to help them to compute. Okay. When you do not have enough dopamine, when these neuron dies, you will not have enough dopamine to support the neurocomputation network, and then Parkinson's disease will arise. So essentially, we know that the typical and defining symptoms of Parkinson's disease are tremor, rigidity, akinesia. These means lack of movement and posture instability. So these defining features of Parkinson's disease clinically, they're actually caused by the dysfunction of the basal ganglion neurocircuitry. So the neurocircuitry that subserves that's responsible for performing the computation that support the movement is not working very well. The reason that these circuitry is not working very well is that they do not have enough dopamine. So dopamine is actually produced in another group of neurons. These neurons then send these messages to control these calculations. So the key for Parkinson's disease is to figure out why we have accelerated death of these neurons that produce dopamine. And through many, many decades of research, it is fairly clear that it is basically an interplay of many genetic factors and environmental factors. So on the side of environmental factors, it is known for a long time. For example, exposure to pesticides, insecticides, heavy metals, so on and so forth, are bad for your health, and they can lead to increased incidence of Parkinson's disease. But more importantly, there are many, many genes in each of us. When some of these genes are mutated, we'll have a much higher chance of getting Parkinson's. Listed in here are 5 genes. When any one of these genes is mutated, you will have Parkinson's in almost 100% of the time. And there are many other genes that have been identified. Mutation of these genes will not in themselves directly cause Parkinson's, but if you have several genes that are mutated, they in combination will cause Parkinson's disease. So these environmental factors and genetic factors work in a way to cause Parkinson's disease in some of us. The exact reason, we're not quite sure. So to understand why we have Parkinson's disease, we're going to focus on one particular gene. That is our research. We're actually focusing on this gene called Parkin, because this is actually a protective gene. So normally, each one of us have 2 copies of this gene. And when both copies of the gene are mutated, we will have Parkinson's. So to illustrate to you what these neurons are, if you look at this, is actually a picture from a rat brain cut this way. This is what we call a sagittal section, right? You can use an antibody to identify these neurons that produce dopamine. So this is actually a remarkable group of cells in that their cell body is actually located in subject nigra, in one part of the brain. And they send very long axons, they send very long process to another part of the brain, striatum. So even on this picture, you can see that it's roughly about 4 millimeters. But what is really remarkable is this discovery in 2009 by a group in Japan that shows that these neurons are truly unique among all the neurons in our brain, actually all the neurons in our body, in that, so they have extremely large axon branches. For example, their cell body is in here. They have a cell body about the same size as many other neurons. They send a single axon without any branches for about 7 millimeters. But once this single axon reaches the target area, it makes massive branches. So the total length of these branches are actually 45 centimeters, so about 13 feet for a single cell that has a cell body about 1,000, about like 100th of the thickness of a hair. So it's a very tiny cell body that makes tremendously extensive axon branches. If you just calculate, if you just do a simple calculation, you can realize that over 99% of this cell is actually in the single axon with gigantic axon branches. So to visualize this intuitively, if you blow up the size of the cell to the size of the city of Buffalo, what's happening is that this single axon is actually roughly to this length about Highway 90 all the way to New York City. And then the more important part is that these axon branches will run all the streets through the 5 boroughs of New York City. So you can appreciate that this is a gigantic cell whose single purpose is to transport this neurotransmitter called dopamine, is transport this message called dopamine to actually deliver it to the target area where neurocomputation needs to take place. So based on this realization, we actually perform a variety of experiments that point out to the vulnerability, the intrinsic vulnerability of these neurons. So in this picture, you can actually see that these green cells are actually dopamine neurons. So these dopamine neurons are from the rat brain, from embryonic rat brain. So we just culture them in a petri dish outside in a cell culture incubator for about 7 days. And even in this highly artificial environment, you can see that this nerve cell, in comparison to the red neurons, which are non-dopaminergic neurons, you can see that these nerve cells have the intrinsic ability to extend very long axons. So we normally do the experiment at 14 days, where the entire field is so green that it's hard to tell which axon is from which cell body. So suffice to say that these cells are very unique. And because of the shape, you can realize that whatever is responsible for this elaborate shape will be very critical for the survival and function of these neurons. And one of the critical elements is actually microtubules, as I'm going to tell you a little bit. So essentially, what we figure out is that for Parkinson's disease, the most critical part is the death of the dopaminergic neurons that is responsible for maintaining the neurocomputation in the basal ganglia, right? So the loss of these dopaminergic neurons is actually caused by dopamine itself. So evolution make a compromise. The neurotransmitter dopamine is a message, but it's also a toxin because we're living in an environment with oxygen. So dopamine is very easily oxidized. When dopamine is oxidized, it produces a lot of free radicals. And these free radicals will specifically kill the dopaminergic neuron, because only in this neuron will you have dopamine, right? So normally, dopamine is actually very highly sequestered. They're squeezed into these vesicles, these little sacs. And these little sacs are moved along microtubules over that long distance, over the intracellular Highway 90, right, to the striatal area for neurocomputation to take place. When you have, for example, good proteins such as Parkin, it can actually stabilize microtubule, so this function is going on smoothly. When you have environmental toxin that is linked to Parkinsons, such as rotenone, it can actually disrupt microtubules. So, for example, when microtubule is disrupted, what happens is that these vesicles will accumulate. It's essentially like if you imagine this is a bunch of trucks that carries a toxic substance. If the highway is bombarded, then these trucks will accumulate in Buffalo. And because the trucks are actually constitutively leaky, so they constantly leak out these toxins. And the leakage is actually causing havoc in these neurons. So that is, in our view, what underlies the vulnerability of these cells. So based on these realizations, we actually have found a variety of ways that we can at least cure the Parkinson-like kind of conditions in a petri dish in these rat neurons. So we can actually use small molecule compound to help to stabilize this intracellular highway to make sure that this transport system is restored. But the problem that we have encountered, and also many other scientists have encountered the same problem, is that it's been very, very hard to generate a model in animals to mimic the human condition. So we cannot really do any invasive study on human subjects. So that's why we need to have animal models to study human disease. However, for Parkinson's disease, it has been tremendously difficult to model this condition in a mouse brain, for example, or even in a rat brain. And we were puzzled for a long time why this was the case. So after we thought for a long time, we realized that one of the simple explanation could be simply the scale of our brain. If you look at this very nice picture, so the mouse brain is actually very tiny. It's not even showing here. The rat brain is like this size, and the human brain is huge. So rat brain on average about 2 grams, and the human brain on average about 1350 grams. For the rat brain, we have concrete evidence. So these dopamine neurons make a massive axon branches. The total length of these axon branches is actually 45 centimeters, right? If you use the cube root of the weight to measure the linear dimension, what's in the rat will become, so in the human brain, what's 45 in the rat will become 4 meters in human. So in human, our dopaminergic neuron is gigantic. This neuron is a very tiny cell, right? It has a gigantic bunch of axon branches. This calculation will be valid with only one assumption, is that we have the same wiring plan between the rat and human. And this is most likely true because we know that all mammalian species arise because 65 million years ago, the asteroid hit the Earth and wiped out all the dinosaurs. So most likely, we have the same wiring plan. So based on this realization, we realize that it is extremely critical to have a human material, to have human neurons to study human conditions. How do we do that? So in 1998, Jim Thompson actually found a very good method that he can actually derive human embryonic stem cells. And with human embryonic stem cells, you can then change the embryonic stem cell into any cells that you like, including dopamine neurons. However, this technology requires human embryos, which is highly controversial. In 2007, Shinya Yamanaka in Japan made a revolutionary discovery. He found that you can actually convert any somatic cell, any cell in your body, into a cell that is for all practical purpose, like human embryonic stem cells. So for example, you can use a piece of skin cells. Okay, you can use skin cells and put in 4 genes. These 4 genes are just human genes. They are normally expressed in early human embryos. If you force them to be expressed in the skin cell, then the skin cell can be converted to what we call induced pluripotent stem cells. So these stem cells have all the characteristics of the human embryonic stem cell, but without the need for human embryo, thus bypassing all the ethical problems. So with these cells, we can then generate human neurons to study Parkinson's disease. So in here, this is actually some skin cells derived from human volunteers. Actually, I think some of you in the audience have contributed to our studies. So for example, we can get a small piece of skin about 3 to 5 millimeter in size, and then from this small piece of skin, we can culture these skin fibroblasts, these skin cells. And from the skin cell, we can put in these 4 genes and then convert them into a cell that is like embryonic stem cell. So these are called iPS cells. So there are actually hundreds of these cells. These tiny cells can be converted in a petri dish into neurons, including, for example, midbrain dopaminergic neurons. So these are the neurons that are degenerated in Parkinson's disease. So with this technology, we can finally have human neurons that are almost exactly the same as the human neuron in your brain. So this neuron have the same genetic composition as the original donor. So with a Parkinson's disease patient, we can then finally have his or her neuron to study with. And more recently, there are even more remarkable discovery in that you can actually convert the skin cell directly to neurons, and also to midbrain, to neurons that produce dopamine. So this is basically the backdrop of our study. And more recently, using this technique, we have been able to find what this protective gene, Parkin, is actually doing in human neurons. So to understand what Parkin is doing in dopamine neurons, we have to look at these synapses. So these are the communication junctures. So these are essentially the little computers inside our brain. So what happened is that this is actually a dopamine neuron. So this neuron produces and releases dopamine. This is the neuron that receives dopamine. So these neurons, they perform neurocomputation to make sure that we can move, right? What happened is that if you look at this neuron that has dopamine, dopamine is actually sequestered in this vesicle to be released in a controlled manner. And once dopamine is released, they're actually taken back very quickly through dopamine transporter so they can be reutilized. But the more important purpose of the dopamine transporter is to limit the duration of the action of dopamine. For example, if you have cocaine, cocaine actually blocks this transporter, so the action of dopamine is prolonged. So people who are addicted to cocaine will have all sorts of problems. So what we have found for parking is that parking is responsible for maintaining the normal expression level of dopamine transporter. So without parking, you will not have enough dopamine transporter. So released dopamine will have, so once dopamine is released, it cannot be very quickly taken back. So the precision of neurocomputation is affected. And on the other side, each neuron will actually release its neurotransmitter, including, for example, dopamine neurons will release its neurotransmitter dopamine in an uncontrolled fashion. These are called spontaneous release, or we can call it leak, right? What parking also does is that it actually controls this leak so that the precision of dopamine transmission is actually very tightly maintained. So normally in this neuron, you release the message and you take it back, so that neurocomputation in stratum can be performed in a very precise way. When you have parking mutations, this precision is gone. And because dopamine itself is actually a toxin, right, it can be easily oxidized to produce free radicals. What we also found is that in these neurons, parking can actually control the level of the enzyme that is responsible for degrading dopamine. So parking can actually set the right amount of dopamine degrading enzyme so it does not produce too much free radicals. So in essence, this good protein called parking is actually controlling the precision of this message as well as limiting the toxicity of this message. So from this, we actually have done further work that shows that idiopathic Parkinson's disease, which accounts for over 90% of the PD patient, has at least some aspect that are similar to parking to parking-linked Parkinson's disease. So with this preparation, we can finally try to think about drugs that can be used to cure Parkinson's disease. So if you look at the current therapies for Parkinson's disease, they're actually geared towards managing the symptoms. You have, for example, dopamine agonists, which is the frontline drug that people use to treat Parkinson's disease at the early onset. So this type of drug mimics the function of dopamine on these striatal neurons, right? You also have L-dopa and carbidopa combination. These drugs actually try to increase the concentration of dopamine in the brain, but in an uncontrolled manner. And you also have MLB inhibitor and COMT inhibitors. These drugs all serve to slow down the degradation of dopamine. So the key is to increase the brain concentration of dopamine. However, all these are in an uncontrolled manner. What we wanted to do is to make sure that you have enough dopaminergic neurons to produce dopamine and to release dopamine in a controlled manner so that it will support neurocomputation in a precise way. And this cannot happen without a very critical component. This very critical component is to find a biomarker. What is a biomarker? If you think about, for example, leukemia, you can easily identify whether someone has leukemia because you have a blood test. In Parkinson's disease, we do not have a blood test or any test for that. When people are diagnosed with Parkinson's disease, they already lose about 70% of their dopamine neuron, so it's too late. We need to find something that can predict that someone is going to get Parkinson's disease, let's say 10 years or even 20 years later. Right now, we're not at that point yet. If we have that, we can then track the progression of the disease. The other goal is that once we have the biomarker, we can actually design these disease-modifying treatments. So what these drugs are, they are actually trying to slow down the progression of Parkinson's disease, or even to prevent the Parkinson's disease from happening. For example, our strategy is to try to mimic the good functions of Parkinson by using small molecule drugs, by using these chemicals that these pharmaceutical companies can make. So if we can find these chemicals, we can then let patients take these chemicals, and then they essentially will help to maintain the health of dopamine neurons. So there is also a high hope for stem cell-based therapy. For example, the transplantation of dopamine neurons that are derived from these stem cells, where we can actually generate induced dopamine neurons. But there are a lot of other issues regarding stem cell-based therapies. So we still have a long way to go, but we already have something that is very remarkable. We have the neurons that are very similar to the neurons in the brain of the patient. So with this technology, I think that it will significantly speed up drug discovery. So last but not least, all this work cannot take place without a group of individuals, for example, particularly Hobo Jiang in my lab, who's instrumental in setting up this technology. And also we have collaborators throughout the world, including at Buffalo. And we got support from Michael J. Fox Foundation and other funding agencies. And the last but not least, I want to extend my special thanks to PD patient. Some of you are actually in the audience. Without your help, we cannot really move our research so far. And it is the concerted help, it is the concerted effort of the patient, their families, and their physicians that enable us to tackle this very difficult disease.
Thank you, Jen. I'd now like to bring up Dr. Gattuso, who will provide a clinical perspective on this, and we will then have a question session, a Q&A session after Tom's talk. Tom?
Well, hello, everybody. Thanks so much for coming. It's an exciting night. I want to thank Dr. Fung for a great presentation and his really fascinating research. I mean, it's kind of sci-fi, I think, to think you can take a cell from an adult and turn it into a cell that is a brain cell. And it's from a patient with Parkinson's disease. It's really groundbreaking. This is the first time we've ever been able to study a brain cell from somebody with Parkinson's disease without having to go into their brain and try and take a cell out, which patients would not be happy with you if you tried to do that. So I've been told I can talk for about 5 minutes, which is good, because as my wife will tell you, who's here, when I get talking about research, people fall asleep. She tells me when she can't fall asleep at night, "Honey, tell me about your research." And that usually works very well. So the title of this session that we're having is New Directions in Parkinson's Disease Research. So I want to talk about a new direction, something that's just kind of been creeping up in the literature, and more people are talking about, and it's getting exciting over about the past 3 years or so. So, we talk about causes and cures of Parkinson's disease. Back in 1817, almost 200 years ago, is when Parkinson's disease was first described, the first patient with Parkinson's by the Dr. James Parkinson. So 200 years, but we still don't really know what causes Parkinson's disease. I hear this from patients all the time. Why did I get it? Why me? And we don't know, is the short answer. In some patients, there's a genetic contribution. We heard about the Parkin gene. But the vast majority of patients are sporadic. We don't really know why they get it. We know that as people get older, there's a higher likelihood that they'll get a disease like Parkinson's disease and Alzheimer's disease. Those are the two main progressive neurologic diseases that we have. What causes the progression of Parkinson's? That is a question that we've been asking for a long time, and we think that over the past 3 or 4 years, maybe we might have a clue as to what causes this disease to progress. You know, when people are first diagnosed with Parkinson's disease, they present usually with a tremor on one side and the hand at rest, some stiffness, and some slowness. And as we heard, they've lost maybe about 60% of their dopamine cells at that time. There's still 40% left. That's a good thing. Wouldn't it be nice if we could protect those 40 cells from continuing to degenerate? But the real problem with Parkinson's, the motor symptoms, we do really well with. The symptoms that are caused by decreased dopamine, our drugs certainly are not perfect, but they do a great job early in the disease, usually for at least the first 4 or 5 years, of keeping the patient doing fantastic, really doing all their activities of daily living, leading a pretty normal life, playing golf, shopping, working. After about 5 years, 10 years, the disease gets more difficult because it's progressing. And the disease moves out of these dopamine cells. It progresses into some other cells that lead to some problems with things like balance. So patients will start having increased falls, maybe have to use a walker or sometimes a wheelchair. And cognitive problems, memory problems, judgment problems, confusion. So these are reflections of the disease moving out of the dopamine system of the brain that we just heard about into some higher parts of the brain. And we've known for a while, let me see if I can figure out how to move this along here, that there is a pathological hallmark of Parkinson's are little inclusion bodies called Lewy bodies. A lot of you have probably heard about Lewy bodies, spelled L-E-W-Y. So these are little clumps of abnormal proteins, mostly comprised of a protein called alpha-synuclein. And patients with Parkinson's disease have these in the brain. What we know is that Parkinson's disease probably starts 5, 10, maybe 20 years before a patient is actually diagnosed with Parkinson's disease. That's pretty fascinating to think that the disease process is starting that far in advance of a person actually presenting to the office with the symptoms of tremor and stiffness and slowness. And actually, patients will tell you symptoms they'll have. Virtually all patients with Parkinson's have trouble with their sense of smell. Sometimes they notice it, sometimes you can only demonstrate it if you do a little scratch and sniff test. But also constipation predates the symptoms of Parkinson's disease. These are kind of non-specific symptoms. When we put them together, there's also a particular type of sleep disorder. We know these patients who have loss of sense of smell, constipation, the sleep disorder, they're at high risk at some time in the future developing Parkinson's. And when you look at the brain in people who don't have a diagnosis of Parkinson's, there's a percentage that have these Lewy bodies in these parts that you can see in red. The one part over here, I'll leave a little pointer so at this point. Right here, this is the olfactory bulb. So this is the part of the brain that controls the sense of smell. And this is the very lower part of the brainstem. So these are Lewy bodies that are appearing in the brain. And patients are asymptomatic. They don't have any symptoms of Parkinson's disease. But what we see is that these Lewy bodies progress. They move. They move out of this part of the brainstem, and they move up into the midbrain, part of what's the substantia nigra, the dopamine cells. And once the Lewy bodies get here, this is when patients start to lose their dopamine cells, and then they start to develop the symptoms of Parkinson's. And so over time, 5, 10, 20 years, very frequently these Lewy bodies then move up into the cortex, into the higher centers of the brain that control things like cognition and, we think, balance. And as these Lewy bodies progress, symptoms progress. Actually, it's a little more complicated than that, but this alpha-synuclein seems to be a real key player. So the question is, why? Why are these Lewy bodies, let me see if I can go backwards. Wouldn't that be great? Oh, yes. Why are they moving? And how are they moving? And if we can figure out how these Lewy bodies are moving downstream, could we slow down or prevent the progression of Parkinson's disease? And this is a new area of research. This is really where you hear about research in Parkinson's disease and when are we going to find a cure and why is it taking so long? Same thing with Alzheimer's disease. This is an area that's really been gaining a lot of steam, is looking specifically at these abnormal protein accumulations and trying to determine What is the process? How are they being transmitted? We know that these proteins can be transmitted from cell to cell. They can move from the upstream cell, move across that synapse that Dr. Fong was showing you in his diagrams, and get to a downstream cell. And if we can determine what are the mechanisms of how these proteins are moving, we think we'll be able to make a significant step towards really preserving patients' futures and keeping them, if they come at the time of diagnosis, keeping them at this stage where the symptoms are just tremor, stiffness, and slowness, and really trying to prevent or greatly slow down the time that the Lewy bodies move into the higher centers. And even better would be if patients who are screened and we see that their sense of smell is starting to decline, maybe they developed a sleep disorder that I was talking about, and we suspect that the Lewy bodies are in this part of the brain, getting them started on some treatments that will prevent it from going from here even to them getting the diagnosis of Parkinson's disease. So that is a new direction in Parkinson's disease research that I wanted to tell you about. I hope you enjoyed it. Thank you for coming.
Okay, we have time for a couple of questions for either Dr. Fung or Dr. Gattuso. And I will bring a mic back to whoever, okay.
This is fun.
Doctor, is there any normal or natural process that will reverse or be able to regrow neurons in this case where the neurons have been decaying and?
So normally we are born with the same number of neurons. These neurons can only die, but there are a specific group of neurons, for example, in certain parts of the brain that can be born from non-neurons. But for neurons that produce dopamine, it is not known that they can be regenerated. So that's actually the most difficult part, is that we do not have a system in our brain that regenerates itself. So so in our body, for example, if you look at those cells that cannot regenerate, these are, for example, neurons, muscles, or actually fat cells. So these are the cells that we are born with a finite number. We can only lose them. We cannot get more of them.
Other questions?
I saw an article recently in the paper out of Denver about work that was being done about a different type of gene using phenylbutyrate to slow the progression of Parkinson's. Are you familiar with that?
I'm not quite familiar with that work. There are so many work being tested on all sorts of compounds that can slow down the progression of Parkinson, like, for example, if you look at a readout in an animal or in a cell culture model, there are a lot of ways that you can see what this actually works. But I think a gold standard is these clinical trials. And these clinical trials, right now, there are no effective drug that can slow down the progression of Parkinson's disease. The only thing that marginally slow down Parkinson's disease progression is these MLB inhibitors, resadgidine, for example, right, Tom? But the effect is very modest.
This is, I guess, kind of related to the question that was just asked. Has Taxol been tried in Parkinson's patients, and if so, what effect does it have?
No, actually, Taxol has never been tested for Parkinson's disease. So for two reasons. One is that it's not progressing to that stage yet. The other thing is that Taxol cannot get into the brain. And actually, various people have been trying very hard to develop Taxol analogs that can get into the brain. These are called taxanes. And people have been designing those drugs to treat brain tumors. And we actually have seen very good response for these taxing analogs in culture models, in these neuron culture models, but we need to move it further to the animal stage and ultimately to see if it works. And if it works, we can try to do it in the clinical stage.
I have a question for Dr. Gattuso. If the Parkinson's is so progressive, why is it that there are days within any one period of time when the Parkinson is very bad and the effects seem to be very bad, and days when it seems very good? How do they account for that?
Yeah, that's a good point, and that's something we hear from patients a lot. It probably has to do with the medications that we use to treat Parkinson's. They have unpredictable effects. And so they have a short half-life, and the dopamine levels will drop in the bloodstream, and then you might have a bad day. And these are, the drugs work the best early in the disease, and over time, things get much less predictable with how well the drugs are going to work, and also you're subject to side effects from the drugs as the doses start to increase. So fluctuations from day to day definitely are not unusual. Yes?
So there's an accumulation of iron in different parts of the brain that correlates with Parkinson's. Is there what's the current view of whether that's cause or effect, or what the role of iron might be in Parkinson's?
Right, so iron has been correlated to progress with a different disease called multiple sclerosis, and just a small number of studies have been done looking at iron concentrations in patients with Parkinson's. It appears that it does correlate with disease progression, but there have only been 2 very small studies done in patients with Parkinson's. We're doing a biomarker study looking at a much larger number of patients and looking at MRI scans. So this is kind of getting at what Dr. Fong was talking about, looking at biomarkers. So we know that intracellular iron is definitely toxic to neurons. It's involved with a certain type of reaction that leads to increased expression of free radicals, which that's the whole oxidative stress hypothesis for the degeneration of these neurons. So whether iron is causative or it's an effect, it's difficult. It's very difficult to say. But certainly it's possible that iron could be, iron deposition, both density and the distribution within the brain could be a biomarker for Parkinson's progression.
Do you know if all of the medications that you spoke about that are now available are available to the Veterans Association at all?
I don't know. I think so.
And would it be possible to get a slideshow or a printout of what you've shown us today?
Yeah, sure. So we can actually make the slide available to anyone.
Okay.
And they're actually making a DVD of this presentation. I'm pretty sure that, I mean, these the currently used drugs for Parkinson's are all very standard. They're available worldwide. I'm sure that they're available to veterans.
Yeah.
Could you comment on the effects of exercise, especially certain kinds of exercise programs running have on Parkinson's?
Yeah, I swear I set you up for that question. So there's a lot. We've noticed for a long time, anecdotally, that patients with Parkinson's who exercise tend to do better over time. There's just been some studies over about the past 5 years, not so much in Parkinson's patients, some, but mostly in patients with cognitive impairment, mild cognitive impairment, and patients with Alzheimer's. And then epidemiologic studies looking at people who exercise clearly shows people who exercise in midlife, 40s, 50s, 60s, who do aerobic exercise are protected relatively against Parkinson's and Alzheimer's disease. There's a lot of animal models. You can kind of force mice and monkeys to exercise and compare them to sedentary animals. But in humans and in animals who you force to exercise, it looks like you get an increase in a neuroprotective factor called BDNF in the bloodstream, which does cross the blood-brain barrier. And we know that this is a protective chemical. So in terms of specifically your question was what type of exercise? Aerobic exercise. This is the kind of exercise like riding a stationary bike, running on a treadmill, doing the Stairmaster, brisk walking. I think the stationary bike, like you're seeing here, is a perfect type of exercise. I recommend it to a lot of my patients because it's tough to fall off of a stationary bike. You hold on and pedal away. Probably about 20 or 30 minutes a day of vigorous aerobic exercise where, you know, at the end of the workout, you're huffing, you're puffing, and you're perspiring.
We do make tricycles for
I think in view of the fact that it's getting pretty close to 7:30, it might be time to call a halt to the proceedings. Dr. Fung has said that if anybody has any additional questions, he can hang around for a few more minutes. So I'd like to thank all of you for coming, and I'd like to thank both of our presenters for a really interesting evening.